Uh, uh, Hi everyone, how are you today? So, how do I sound? How do you hear me? Is it okay through microphone? Uh, it's a shame that I forgot bringing my face shield today, so I cannot take off my face mask today. So instead, actually, I am wearing double layers of face mask. So I still hope that you can hear me all right. So if that's then fine. So today I'm going to use this microphone to deliver this lousy lecture. So last week we looked at the basic components of nucleic acid. So like we know, assuming that nucleic acid include only two members, which is DNA and RNA, and both of them are actually fairly important material, because we all know that already. The DNA is very important because it serves as the genetic material. And what about the RNA? RNA is, right now actually, RNA plays something like a second fiddle, like playing secondary role, on less important than DNA, but nonetheless, actually, RNA is, actually, to tell you the truth, when it comes to the origin of life, the RNA began the life first, for some reason. I don't want to go into the detail over here, but that's the scientist consensus in these days. In the first prototype of a living cell, DNA cannot. There's no way DNA could be the genetic material for some reason. But RNA could. So and there are lots of evidence actually support the RNA. Not only RNA could behave, act as a genetic material, but also RNA is very versatile. So it can play many roles at the same time. So in the beginning of the whole life, RNA had to be the genetic material. So and not only that, RNA actually shows tremendous different roles. So you will be actually surprised if we go into the real details about what RNA can do. So actually the DNA is the one very boring material, like we said, the other day. But the only good thing about DNA is very reliable, very stable in chemistry. And that is actually very important. If we can use DNA as the storage, the sacred place to trust our entire thing. DNA is everything, because if we think that our genes are everything we need, as long as we have genes, then we can always start over. If that's the kind of philosophy that we agree on, then DNA is the only thing that we need to keep. For that matter, DNA is very stable chemical structure. It's the one actually we can fairly rely on. So that's the really good thing about it. But other than that, actually, DNA has a lot of those the flaws in terms of how many different things can you do. If we ask DNA, then actually not much. But RNA is different. RNA is a kind of cousin. RNA is a cousin of DNA. As we already saw, what's the difference between if the basic fundamental component making up either RNA or DNA are so similar like this? Remember last week? DNA, what does it take to make DNA? You need the individual component. What's the name of that individual component? That was nucleotide. And what's the component, composition of the nucleotide? Yeah, it has a three part. One is you need to have some kind of a sugar, ribose sugar, 5-carbon sugar. Both RNA and DNA has those ribose sugar in their respective nucleotide component. The only difference is in the ribose, they do have still this oxygen in this particular position, whereas in this particular position, DNA, for some reason, this oxygen is missing. Okay? So that's the difference. And that actually made a tremendous difference, actually, in terms of a chemical stability. And that is the reason why, eventually, we decided to kick them out. We used, in the beginning, to ask RNA nicely, could you become our genetic material? And then RNA say, sure. And then RNA did all those marvelous job. And then, for a while, after we settled down, okay, hey, why do we have to keep this guy as troublemaker? Because it is chemically so unstable. Because of this, this oxygen. Oxygen is always a troublemaker. And this particular position of oxygen makes this RNA is a long chain. It's so chemically unstable. Okay? So in these days, actually, whenever we deal with the RNA in the laboratory, that is actually the pain in our behind, because RNA we cannot let the RNA sit on any room temperature. But then in a matter of a minute, it will degrade themselves because it's chemically very unstable. But DNA is extremely stable, comparatively speaking. So that is actually tremendous advantage if we use either one of them as our storage of genetic material. And that's the reason why. But other than that, actually, nothing else actually is something that's very different. Both DNA and RNA, it has one position, either one of these, nitrogen base, either one of these, okay. Both RNA and DNA also has this one particular part we call the phosphate group. So basically, they are very similar molecules. That's why I can call them as they are cousins, kind of very similar molecule. But still, DNA acts as a genetic material, and the RNA in these days kind of acts as a copying, a running errand. It is trying to copy the information written and stored in the DNA, and then get the exact copy, and then deliver themselves to the site where the protein synthesis is occurring. Hey, it's me. This is the message I got from the other guy, the gene. So why don't you guys read me? Can you, if you can? And there are some structures in our cell who can do that. The name is ribosome. Ribosome can read specifically. Ribosome can understand what's the meaning of all jobbles of the so-called DNA sequence or RNA sequence. The sequence. What's the sequence? When we casually refer DNA sequence, why is it so important? Have you heard of this so-called genome project? That was a huge project to read all those DNA sequence in human cell. Okay. How many nucleotides in our DNA do we have? We have 6.4 billion nucleotides in a single cell in a human. 6.4 billion. And they exist in pairs. So in other words, actual number of pairs is a 3.2 billion pairs of nucleotides. But how many different kind of nucleotides can you have? You have only 4. You have only 4 different nucleotides. And you have all these seemingly totally not making any sense jumbles of repeats of either one of random repeats of these all different 4 nucleotides, and they are connected together. It's a DNA sequence. And why do we want to read all those DNA sequences? It was a really, really important project. It took 10 years. It began in 1990, I guess, and then it ended in year 2000 in the United States. Yeah. Billions of dollars, US taxpayers' dollars, poured in. And they really, really need it. And then, thanks to that, actually, in these days, we are facing totally whole new world with the tremendous, oh, where is my microphone? With the tremendous potentials. It changed the whole things. It's a very scary future because of that nowadays. So, I mean, we don't really go into that further detail right now at this point still. But anyway, DNA sequence means simple order. A or G, C, A, G, 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 T, T, A, A, T, T, C, C, G, G. What about this? What does this whole thing mean to me? I don't know. I don't get it. But there are in the cell. There are some structures who can read, who can understand this meaning. Why? Because those random, seemingly meaningless random sequence, actually, a lot of times it is really, really something because it can determine. It can determine what kind of protein will be made. It is kind of a secret code. So there is a reason why a lot of times we call this as a genetic code. DNA is a genetic code. Something like a Morse code. You do this, but somebody can understand. So certain, at least not all, but certain area of this our DNA holds some segment area can provide. Okay, here is the thing, the instruction. That's the reason why I compared last week that DNA serves as some kind of a recipe, the cookbook, the entire whole cookbook. Each page describing what kind of dishes can you make if you follow this recipe. In this occasion, the recipes is for making each different protein. Because protein is also nothing but a whole different array of 20 different amino acids. Let's say, for example, let's say you have one particular protein made of 50 amino acids. Then how do you make that protein? You are given a choice of picking randomly 50 amino acids in sequence in a row, but you have 20 different choices. So position number 1 in an amino acid, you can choose 1 out of 20 amino acids. Okay. And the second position, you can also choose another amino acids out of 20 amino acids. And you can also repeat the same amino acid. So you have so many tremendous possibility of generating certain particular sequence of amino acid. And because of that, you can generate actually tens of thousands of different kind of proteins, because protein is nothing but whole mixture, assemble of amino acid, and what kind of component as an amino acid come in, and which particular position you can generate different protein. And the length, I mean, it's not limited also. Like, for example, like how many amino acids can I have in a given protein? As many as you want. As little as you want. You decide. So you can make protein made up only something like a 20 amino acid, very small protein. And also you can make, in some other occasions, make a protein consisting of 1,000 amino acids. That's also possible, and that's also exists in the cell. So if you were like something like that, then your possibility is endless. But in reality, your possibility of making different types of protein is limited actually by who? The DNA. DNA will decide what kind of protein can you make in a cell. How? Because the DNA sequence, this DNA sequence is the one actually you have to follow. If you want to make a new protein, you cannot start from scratch. But the way you make a protein, you always have to start from certain DNA already instruction encoded. And how do you get access to that information? Through RNA. RNA will copy certain region, segment, as a gene. That's what we call gene. Then those copied information will be delivered to a specific location in the cell. And those little guy, ribosome, it's not little, but this snowman like a ribosome, can actually read those each individual sequence of order. Sequence means order of nucleotide. Order of this nucleotide on the RNA. That is simply just being copied from originally in the DNA. And then the meaning of each different sequence of those RNA is because each different segment of the sequence will actually signify what kind of amino acid will come. So this ribosome is the one, that is the reason why ribosome is the one who make the protein synthesis. Because in the ribosome can call up some different amino acid to the site. Hey, amino acid number 1, come over here. I got an instruction that needed you. That's what the ribosome does. And the second position, this ribosome read, this sequence. That means another amino acid must come over here, and then come over here, and then ribosome will click and link these two amino acids together, and one by one it keep going until the end of this protein synthesis. I don't know if you didn't take any of this biology course before in your entire life yet, then still, although this is a kind of a really the elementary as possible version of explanation that I can may think of about what's going on in the relationship between DNA and RNA and the protein, but probably it will take some time. If you didn't learn anything like this in your life, but it will soon, you will be familiar with. And then it's actually, it's nothing. It's not really that difficult one. So, having said that, so each of this nucleotide, and there is a reason why DNA sequence is casually in your TV series. For example, the crime scene, detectives will try to read some of those DNA evidence, or then a lot of times actually the information comes with the A, C, G, A, T, T, C, Z. Something like, what are these? These are individual, the abbreviated form of each different nucleotide of the sequence. So A means the nucleotide having the adenine. Where are your adenine? Here. This one. C means the same thing. Everything else is the same as far as the DNA nucleotide goes, but the only uniqueness comes from this part. In this occasion, here the C means tyrosine. Okay, This cytosine nucleotide is the one. G means the guanine. So then in that way we can simply abbreviate all different type of nucleotide by one letter or alphabet. And that's what you are reading. Because only this one letter identify is the whole different types of nucleotide. And same thing goes for true in the case of RNA too. In the case of RNA, the only difference is what? You tell me. Compared with DNA, what is the only difference in RNA? You don't see this T here, and instead of this T, you substitute this T with what? U. That's it. So, on the other hand, in all these sequences, if you get to see somehow a U, presence of a U, then you immediately know, oh, this must be an RNA. On the other hand, if you see T in the sequence, and then you automatically know, oh, this must be a DNA sequence. That makes the difference. Okay. What about this? Can you for a moment examine this particular structure? So now last week we covered all different kind of sugars and lipids. So you probably now got sick and tired of seeing different chemical structure already. So that means you are expert already now, chemical structure. So I should be able to ask you to examine this chemical structure. What does it look like? How do you? Is it carbohydrate? What are you going to do if I actually ask this kind of a question on your exam? Tell me, is this carbohydrate? No? Why not? Yeah, answer is right, but why? Why is it not carbohydrate? Because a certain ratio doesn't fit. If it's going to be a carbohydrate, carbon and hydrogen and oxygen, there's got to be some specific ratio. Carbon, hydrogen, oxygen, certain ratio of, but if you count the number of carbons and oxygen, and then you don't really have that fitting. So this must not be a carbohydrate. And is it lipid? Why not? Is it easier to determine than carbohydrate? Yeah, it is not a lipid, no. But how do you know? Why is it not a lipid? I mean, that is actually the purpose of that we went through in last week, like, so that we can see something, then, oh, it must be this. This is actually the purpose of going through all these 4 different macromolecules in life. But obviously, this is not a carbohydrate, and this is not a lipid, because you don't have any hydrophobic part, non-polar part. Everything, almost everything is actually polar, extremely polar. This means a phosphate. This P stands for, actually, this is like shortened form, but this P stands for exact the same phosphate over here. Why this one is not following? This is the P that you saw the other. Okay, now it's. So this is not a lipid. Then, unfortunately, we didn't cover the protein yet, so there's not enough information we can use whether it is a part of a protein or a nucleic acid. But one clue is nucleic acid consists of what? Nucleotide, right? And nucleotides, individual nucleotide is made up of what? 3 different parts. Remember? One part is sugar, ribose sugar, right? And another part, nitrogenous base, which is adenine in this case. Oops, okay, something, we are getting something. And the other part is phosphate. But the only difference is we're supposed to have 1 phosphate here, but unfortunately you have a 3 phosphate. So that is a little bit of a disqualification. If you didn't have this, okay, out. I would love to call you immediately, you are one of the nucleotide, okay, fine. But these guys, actually, you know what? Actually, when all of these nucleotides are going to get into the long chain of like nucleic acid being synthesized, the kind of form that enter that synthesis is not this, but these 3 phosphate containing form. And during the entry, they have to clip these 2 phosphate off. And that's the reason why in the living existing, whether it is a DNA or RNA, all these nucleotide, you have only 1 phosphate version of a different nucleotide, but in reality, when they are being used in actual synthesis, they have to enter in this 3 phosphate containing form, energetically speaking. Do you still remember this? The hydrolysis, the chemical reaction, energy releasing reaction, spontaneous, and an energy requiring non-spontaneous reaction. Certain reaction is something that can occur spontaneously. Okay. And some other reactions cannot take place spontaneously because it is against the rule of nature. Okay. Whenever you try to assemble, you still remember this hydrolysis and the condensation reaction last week. The condensation reaction, you are trying to build small component to assemble the bigger molecule, right? In the condensation reaction. Do you still picture that condensation reaction diagram or totally forgot about it? Condensation reaction. That the one water molecule is out, but you're trying to assemble, connect 2 small components. Whether in the case of this carbohydrate buildup or lipid buildup, you always have to go through this condensation reaction, wasn't it? Yeah. That reaction. So try to picture that reaction. What's happening is you have 2 small components, then you are trying to connect these 2 little components to result in a larger component, and then you are keep going on. So tell me, is it going to be a spontaneous reaction? What do you mean spontaneous? Spontaneous reaction means if you let them sit over there, but they will simply occur without any intervention because it is supposed to happen because it's a natural way. Okay? And the answer is that kind of reaction is non-spontaneous. It is non. Why? Because it is against the nature. The nature always tries to break things apart. So nature from the big molecule, breaking the big molecule into smaller components is a spontaneous reaction. Why? Because it will release the energy. And it is about the entropy. Have you heard of the entropy before? entropy is something, the kind of status, how disorderly as a little condition in a given location. And the second law of thermodynamics states that the entropy of the universe will always keep increasing. Like in this room, our entropy is fairly low because we are all ordered, sitting nicely. But in order to increase the entropy, what can we do to increase the entropy of this room? Everybody moves, stands up and move around like chaos, crazy, and then go out. Then our entropy will be increased. Which one do you like? The latter. It's a human nature and the universe nature. So universe will always favor this increased direction of the entropy at the end of the chemical reaction. It will occur spontaneously. You don't really have to do anything special, because if you let them sit over there, they will, without doing anything, in itself, it will happen like that. That type of reaction, we call that a spontaneous reaction. So to simplify, to determine whether certain reaction is spontaneous or not, it's easy, because anytime you try to break a complex molecule into smaller parts, it is a spontaneous reaction, because imagine the end result of that kind of chemical reaction. If you break the big molecule into the little pieces, and then all those little pieces will be scattered around, right? Then do you get to increase the entropy or not? If you make those things little scattered away, then the order will be decreased. It will become more disordered. So the entropy will be increased. So is it spontaneous reaction or not? Still not sure? If you make things more disordered, is it spontaneous or not? Spontaneous, yes. Why? Because now you increase the entropy of the system. The backward of chemical reaction, think about it. If you had to pick all those scattered components and then put them into a really nice little organized structure to build a macromolecule, right? Then as a result, do you increase the entropy or decrease the entropy? Initially, you were sitting like this, all scattered around. But then if I call you up and then let you guys stand in line so nicely in this little podium, then did I increase the entropy or decrease the entropy? Decreased, yeah. So is it spontaneous reaction? No, it is not. That means universe does not bless you doing this kind of a reaction. So in chemical reaction, there are only two different kinds of reaction. One is spontaneous and the other one is non-spontaneous, depending on this status of entropy. Entropy, always you try to think about how disorderly it is. If the system is more disordered, then it is a higher entropy. If this whole system is more organized, okay, taking up smaller space because of this higher organized status, then it is a lower entropy. Actually, the cell, extremely complex and then highly organized structure cell it is a very low in entropy cell itself, right? But the problem is, in universe, you're not supposed to have any, this is a highly organized cell spontaneously. And that is really, really one very good point that the creationists always try to attack. Hey, What's going on? You stupid scientist, you don't know? You don't even heard that the law of thermodynamics, the second law of thermodynamics, entropy? You're not supposed to have this cell spontaneously because it is totally against. You are violating you guys' own scientific, the fundamental golden rule. So there must be a creator God who has done this. Is what really those creationists love to point out this law. Seems paradoxical. But in reality, no, it is not. Because just because it is a non-spontaneous reaction, that doesn't mean there is no way you can do this. Just like everyday our lives. When you are encountered with some kind of a difficult task, hey, this is some kind of against our system or rule, so you're not supposed to do this. Then what do? Take out your wallet and then try to bribe them. Then it usually works. Usually. Yeah. So is the nature's system. How do? How do you bribe in nature, in chemical reaction? You provide energy. So if you provide the energy from outside, then you can drive this non-spontaneous reaction. So, in other words, in order to generate this cell, like highly organized structure, somehow the energy must have been provided into this, and actually the procedure involves the further more increase in the entropy, the other side. So overall, because this whole law states the overall status of the entire universe, but we are only looking at the very local area. If you wanted to, and then decreased local area of entropy somehow, then because of that, you had to increase further some other area. That's the whole thing. There is no free lunch. Because that's what the first law of thermodynamics states, actually. There's no way you can create new energy. You can only change the form of the energy. And actually, that is another very important point that we need to go throughout this whole lectures. So anyway, that's the thing that I need to make you understand this spontaneous reaction versus non-spontaneous reaction. In other words, the point is, in non-spontaneous reaction, you need to provide the energy. And the vice versa is true. If you perform any spontaneous reaction, what's going to happen? Energy will be released as a result. And that is the reason why you are having meals every day. You are conducting the spontaneous reaction inside of your body, starting from a big chunk of a molecule, like pop, 떡, ramen, whole chunk of food molecule, and what you are doing is what? Breaking them down. So, this example of non-spontaneous reaction I gave you was trying to assemble little piece of a component into the larger one is non-spontaneous because you are generating a more ordered, less entropy molecule. Then think of the other, totally the opposite way of a chemical reaction, starting from a big molecule and you are breaking them down into little pieces. Then all those little pieces inevitably will be scattered around, increasing the entropy. So it is a spontaneous reaction. And good thing is, chemical energy stored in the big body will be released. When they were were broken into little pieces, the chemical bond what we have learned, in particular, that covalent bond, they store the energy. Why? Because in order to generate chemical bond, you need an energy. Because establishing the bond is a non-spontaneous reaction. So how can you make this thing happen? Put the energy. So that was the thing. Then if you want to reverse this thing, then the energy stored in the chemical bond now will be released. So that's the whole cycle of this thing. So you are doing this. Releasing the energy. Why? To use those energy for your own need. That's what you do. That's the reason why you are eating. Why do you need energy in your body? Because there are lots of business that you need to take care of. And a lot of them actually, the business that requiring the energy. You need to build some material. Whenever you try to build, then you must provide the energy. So where are you going to bring those energy? That's when you come up with this. You are encountered with the first law of thermodynamics. Hey, unless you are Almighty God, there is no way you can create the energy, nor can you destroy or spend any energy. Energy is still out there. The total amount of energy in the universe does not change. The only thing you can do is simply convert the one energy form into different form. So you are only borrowing the energy originally stored in the food molecule, simply breaking them down, so release the energy, and then trying to convert this energy into something else that you are needed. In those, why am I getting into this total messy business? Because of this. Jesus, I shouldn't have started this. But, so, this is not, where was I? This, why in building up nucleic acid, like whole chain of different nucleotides, whether in the case of DNA or RNA, the reason why you need to have this molecule having this 3-phosphate instead of single phosphate is while entering, these 2 phosphates are clipped off. So in this way, you are performing a spontaneous reaction. Because if you break this thing, energy will be released. The cell is so clever in that. Why? Because think about it. Building up, assembling the DNA. Is it spontaneous reaction or non-spontaneous reaction? In order to build the chain of DNA, you need to assemble, pick each different nucleotide and then assemble together. Is it spontaneous or non-spontaneous? Non-spontaneous. So you need to bring the energy. But this clever guy, the cell, what they do is use individual this nucleotide, and when they try to come and then being inserted, hey, you guys need to clip you guys off and then come in. So by doing so, this position, energy will be released. So use, hey, this is my entry fee. Okay, I got energy, so you can come in. So they don't need any extra energy, but by employing this kind of a strategy, they take care of this energy requirement and then able to assemble whole chain of this nucleic acid, whether it is an RNA or DNA, same thing, starting from this. So that's the thing that I needed to explain. There's another thing that I need to explain using this figure is more important thing. The name. Oh, yeah. Is a nucleotide, is it? And this name is what? ATP? Have you heard of any ATP before? ATP is just kind of like the symbol of energy in the cell. ATP, another way of call ATP is probably I can say is an energy currency. Anybody economics major over here? Energy currency, the money. So instead of we try to trade each of different material, just like our economy system, these guys use some kind of more convenient form of the energy transfer. So that's why this ATP can be compared as currency, energy currency, the money, like cash. A lot of times actually in general biology book, this ATP, they compare this ATP as energy cash. So whenever they need some kind of energy, well, you could imagine that, oh, I need to put some energy. Then, hey, come over, glucose, come over here. And this big glucose will come over here. And then, why? What do you need? And, okay, I need some energy. So I need to break your, the hands and the legs. So you break all these different chemical bonds of glucose and the energy will be released, okay? And then you grab those energy and then use it. That's one option. Instead, you don't do that. Instead, you do those breaking glucose business somewhere else, and then by collecting all those energy released from glucose, then you immediately convert those energy into this ATP. So it is so convenient, like your money in your wallet. And then whenever you need some energy, then you provide this money over there. Hey, ATP, go over there. Then ATP will work as an energy. How? How do you actually practically use this ATP as an energy currency? Exactly doing same thing, this. You break this thing. Then the energy will be released. And you just clip one phosphate off instead of two in the case of the building nucleic acid. So then the end result will be if you clip off this phosphate in the ATP, then it will become what? What did you say? Yeah, exactly. Thank you. So you took the chemistry or biology before, huh? No? Yes. ADP. So here this D or T means tri versus di. ATP stands for adenine triphosphate, 3-phosphate, whereas ADP means having only 2-phosphate, ADP. The relationship between ADP and ATP is, that's the point that I need to make, is your rechargeable battery. Your fully charged rechargeable battery is ATP. And then you use battery for your energy, then it becomes used battery, which is ADP. Why did I compare this as a rechargeable battery? Because ADP can be recharged into ATP. Simply, this is a cycle. How? You simply put this 1-phosphate back into. That's how you synthesize ATP. And so you keep cycling this ADP versus ATP, and that's how you manage your money, your energy in the cell. And whenever you need this ATP synthesis from ADP, then you need to break your food molecule down. So those energy will make possible to assemble the ATP from ADP. And whenever you need to spend the energy for whatever business you need to, then ATP will go over there and then sacrifice themselves by clipping 1-phosphate off and they provide the energy. If you take this 1-phosphate off, then energy will be released. So you are using, just like you are using this any battery, then now those once ATP now will become, after after releasing the energy, will become ATP. And then go where the food is being broken down to recharge themselves. That's how you do it. So this is that ATP, the energy. Everybody. It's so marvelous in that no matter who you are, whether you're the virus even you're a virus or bacteria or human, plants, no matter who you are, everybody use this ATP as this kind of energy currency. That's amazing thing. Right? Even in the whole world, people don't have 100% agree in what kind of currency we are going to use worldwide. So majority of us were trying to use the dollar, but some others will try to maintain and use some other currency like the Chinese currency or whatever. But here in the living system, they don't. They always use this ATP as only strict current system. Okay. And the point is, this is actually a type of nucleotide. It is an RNA nucleotide. Exactly. So this one is a kind of a dual role. Not only can it be used as an energy currency like ATP, but this actually the exact same ATP can also be used in assembling RNA. It cannot be used in the DNA assembly, unfortunately. Why? Because it has this. But if you have this thing removed, then of course this thing will be able to be incorporated in the DNA generation. So that's why are you acting up to like this? Another thing is, now we are moving so slow. Another very unique thing about this nucleotide is their very peculiar, unique way of forming chemical bond. And with that, we have a kind of thing called this complementary base pairing. It's always between one adenine and a thymine, the nitrogenous base, and only between guanine and tyrosine. And as you see over here, two lines are drawn between A and T, and three lines, solid lines, were drawn between G and C, means the different number of hydrogen bond. So this chemical structure, here the letter G stands for this is a guanine. Nobody needs to remember the chemical structure, exact chemical structure. So, and this one is the tyrosine. And between guanine and tyrosine, as you can vaguely recognize, all this dotted line indicates the hydrogen bond. Three hydrogen bonds are possible and they fit in. On the other hand, over here between A and T, only two hydrogen bonds are possible because of their structure. Yeah. So it's just like an electric plug. Like here in Korea, you have the two holes electrical plugs are needed, but if you go over there in the United States, and then three holes plugs, so it doesn't fit like thing. So between A and G, there's no way they can form hydrogen bond. No, and same thing between T and C or anything else. Only this hydrogen bond can be formed between always only A and T and G and C. So that makes some kind of a special already discrimination, and that turns out to be extremely, extremely important in whole life. So now that's the reason why it's named as a complementary base pairing, because complementary means only some of the unique, special relationship. The good thing about this complementary base pairing nature is now we can automatically know, let's say in the case of DNA, we know, assuming that we all know that DNA usually exists in a two-strand, a double strand. The reason why they exist always this double strand is because of this complementary base pairing thing. And if you, like, let's say if I gave you only one DNA sequence of these two double strand DNA, if I gave you, hey, this thing should work. This one is why it's acting up like this. I don't like you. So for example, like AACAGAT, this was only one strand. But I don't have to give you a full strand of the double strand. The minute you see, then you automatically figure out, you can figure out the other part, the sequence, because of this already complementary relationship. Automatically you, T, T, G, T, C, T, A, like, voila. And that is, it's not for only our convenience, but there is something very, very significant in that. Let's try to imagine, maybe, I'm not really sure that is a very good example or not, but I will just go ahead and do it. I don't care. Let's imagine that you're trying to do a makeup. Okay? I saw a lot of ladies actually doing those makeup in the morning, the commuting subway train. But what is the most important thing that you need to do? You need to make the completion of this makeup. You cannot do this thing without this one item to do this makeup. Oh, right, hand. Exactly, yeah, but assuming that you are lucky enough to have both hands, yeah. And what else? It is non-living item. Yeah, can you do it without mirror? Well, if you can, then there is no argument can be made. But I thought you guys cannot do this makeup without mirror, usually. So let's say that is correct, my assumption, and then go ahead and complete my argument. So without the mirror, you cannot do this. And this thing is exactly serving, this complementary relationship is serving kind of mirror-like things. So what can you do? They can assemble, they can create their own copy because they know these complementary base pairing things. So when they try to do this replication, why these replications are so important? Remember, DNA is the uses, the beauty of, the reason why we regard DNA is just like a God sends, is DNA can act as a genetic material. What's the most important thing for any genetic material? It should provide us a whole longevity issue, should be able to make us keep going forever and ever through what? Reproduction. And what do we do during the reproduction? First thing to make this reproduction work is you have to double up your whole genetic material. Without it, you will be miserably a failure. Like, whole this reproduction means from original one, you have to split into 2 copies. This is how you do in your cell division. This is the basis of reproduction. But then, let's say have you 100, for example, the amount of the genetic material, 100. gene. And you simply, without taking care of this replication first, then you equally divide this thing into 50-50, and this equal have 50 will or be totally a failure, disaster. So what do? Just right before this cell division, you always have to do this replication. Temporarily, you increase you into 200, and then each 100 will be inherited into this. Now, each resulting, your offspring don't have any problem because you inherited exactly a copy. So, for that matter, this DNA replication is extremely, extremely important. But also, that's where you create a lot of mutation. That's why you are here having, each of you have a very unique trait. So this is in both ways, it is one of the most significant event actually in our continuity of life. You just provide the stable continuity because of this replication, and also inevitably you create a very variable trait at that point. During this DNA replication, you are supposed to have there is no way you can stop it. You can avoid. Mutation will occur, no matter what you do. And that mutation is the source of your genetic variation. A lot of actually those mutations are not good, not desirable. So those unfortunate ones who bear those mutations will suffer and not going to be able to make through their lives. But only for those who are lucky ones, somehow obtained some very beneficial mutation, will be able to expose and then exhibit the new feature or new trait. And that's how we are now. We end up whole different individual with different traits. So anyway, so this complementarity is the source of the convenient DNA replication. No other material will actually be able to provide such a convenient way of copying things. But each of two DNA strands can serve as each of this template. Some kind of example was standard. So when this is the way they hold up these two, each nucleotide in DNA double strand. And each of them have unique complementary base pairing. So when they up for new copying or during the DNA replication, all they gotta do is split up the existing two strand. And for each existing strand, new nucleotide will be added on automatically according to this complementarity. Here, the mirror analogy will take into action. You don't really think too hard, and you don't need any special template providing device, but each of these strands will serve as a template, and all you gotta do is hey, enzyme, I know your enzyme are kind of stupid, but at least you should be able to do these things, and they can. So all they do is, this, if there is a T, then I need to bring an A, and then insert. And if there is a C, then I need to bring this G, and you complete this whole business. And then all of a sudden, whole new two sets of DNA strand will be made. Then that's how you do this so-called DNA replication. Okay. Yeah. Jesus. Why am I moving so slow? Okay. So that's, although there are so many different things that I can talk about this DNA and nucleic acid and RNA, but right now it's only, it's not really appropriate to go into further details. So let's move on to the next and the last topic of this chapter, which is proteins. So protein is a polymer of amino acids, just like we can say about DNA or nucleic acid is a polymer of nucleotides, right? And fats are polymer of fatty acids. Polysaccharides are polymer of monosaccharides, just like that. So that's the whole relationship. Always, these whole macromolecules are individual components assembled together. So right now we can have some kind of a big statement about whole one particular characteristics of all these whole polymer building things. It is all what? Is it spontaneous reaction or non-spontaneous reaction? In building up all these 4 different types of macromolecule, what kind of chemical reaction is this? Spontaneous or non-spontaneous? Yes, at least that one, if you can have a little bit of a more comfortable thing about, then that will be just good enough. So all this, to build up these different polymer, different macromolecule, every different types of a chemical reaction with all will be non-spontaneous reaction. That means energy. You need to provide some of the energy. What kind of energy? In what form can you bring them? Practically speaking, in the cell. So now you agreed that to synthesize all different types of chemical, these macromolecules, you need energy. What kind of name the kind of energy that you need to use in the cell? Yes, ATP. That's where ATPs are all needed. Yeah. Where do you get the energy? Those ATP. Who gave you the ATP? Your lunch. Or your supper, dinner, if you had the early dinner. How? By breaking your dinner down, and then energy released, and somehow you convert those energy into ATP. From what? ATP. That's all. Now, protein is a polymer of amino acids, and connecting each amino acid to build whole array of assemblage of amino acid with the name of a chemical bond we call as a peptide bond. And there is a reason why sometimes instead of this protein, you get to see this word polypeptides, which is synonymous to protein. A lot of times. So either polypeptides or proteins, and a lot of times they are the same word. Sometimes protein does not mean a single individual polypeptide. Sometimes four different parts. Actually, it's a very good example is hemoglobin. Hemoglobins are 2 alpha and 2 beta chains of polypeptides are gathered together to assemble one functional hemoglobin protein. In this case, what we can, is there any different color? Because this iPad is not working. In this case, each part is one polypeptide. Because this is one individual assemblies of amino acids into, actually, if this hemoglobin works as a single, so-called monomer, then one polypeptide would be equal to one protein. However, in this occasion, four such different polypeptides, which are actually each of them a protein, has to be assembled together until actual functioning, really functioning unit of hemoglobin protein. In this case, we only call this whole entire four different polypeptides as one single hemoglobin protein. So only that type of special occasion, there is a difference between polypeptide and protein. But other than that, not everybody has this kind of occasion. A lot of times, only single protein work as a solo. In the case, one polypeptide is equal to one protein. So, just in case, if you are wondering, I'm not going to. Of course, you don't have any business to having to remember this kind of thing on your exam. That is not the point. That kind of little detailed information is unnecessary. One. And this one is also you don't need to worry about. But if you are actually, this is one of the always fundamental steps everybody has to take when they are about to learn the protein, because this is a basic structure of general amino acid. Amino acid is always one carbon, one central carbon. We call that as an alpha carbon, around which you have a two part. One such part, its name is amino group. That's why it is called amino acid. One nitrogen and two hydrogen, who cares? The opposite side, always you have a carboxyl group. Its name is a carboxyl group. And it always has these two requirements. Every different amino acid must have this. And one other, you have the one hydrogen. So anything, any chemicals that can show this arrangement can be regarded as an amino acid. Yeah, everybody. The only difference is here. Remember, carbon can have four. Carbon can have four different covalent bonds. So it has used up one, the other one over here, and the other one over here. Still, this carbon has one more carbon, the covalent bond left here. Okay? So this is where the uniqueness of any different amino acid can be played. This R can be anything. Any different chemical structure, crazy chemical structure can be fit into over here, as long as they can whole as a whole, as long as they can form a covalent bond with carbon, which is not a difficult one. So that means you can have so many different amino acids. So many. In nature, really existing. Maybe hundreds. Several hundreds, maybe, if you carefully look. But among them, only you have 20. Such particular amino acids are the one participating in your protein building structure. So if you examine in your protein in your body, okay, if you examine protein, all different protein, you have, it's been said that you have about 100,000 different protein exists in your human cell. So if you examine each of 100,000 different protein and then determine the amino acid, what kind of amino acid you have, it's either one of those 20 amino acids. But that doesn't mean that there are, in the whole our living system, there are only these 20 amino acids. No, there are so many more for some reason. Okay. Actually, and also another thing is, some bacteria, so it's kind of a general consensus that you have only 20 amino acids in protein. But then somebody else will say, no, it's not. There are 22 different amino acids in the protein. What? What do you mean? Some bacteria, it turns out that recently it was discovered that some bacteria have 2 more rare amino acids incorporated in their protein system. It's a newly discovery. But anyway, so it's not entirely 100% accurate to say that there are only 20 amino acids in protein in our living system. Depending on who you are, you can have a 22 amino acids. Okay, although you are not bacteria, and more weird thing in these days is some crazy scientist, actually they have a synthesized artificial cell, artificial bacteria, where some whole new synthetic protein can be incorporated by creating whole new genetic code. So they are doing all these different crazy things like that. So with that, I will continue more about this protein things made of 20 different amino acids. With that, we are going to have a 20 minutes break. And was it 20 or 25? 25, okay, yeah. 25 minutes break. So we will start at 6:40, the second session. Okay.